after 9/11, several letters were sent out to journalists and congressmen and senators on the East Coast, and it turns out they contained a weaponized version of the worst strain of Bacillus anthracis. Back in the 1950s in Texas, the most deadly strain of Bacillus anthracis mysteriously appeared. It was studied at the University of Iowa. The CDC had a center there in Ames, Iowa, and so it was named the Ames strain. Ames strain really does not occur naturally, and it is the most deadly form of Bacillus anthracis because it has two plasmids on each genome. And if you kill the host cell of that genome, the host cell of Bacillus anthracis, these two plasmids join together and form a toxin which is deadly to humans and animals. So right after 9/11, the FBI came to our lab at NAU because Paul Keim had just several months before that published a paper regarding looking at the genome of Bacillus anthracis and being able to break that genome into different strains. No one had ever done that. No one had ever cared. I happened to be the head of technology for the College of Arts and Sciences at NAU. Paul had a very small lab under the College of Arts and Sciences. Suddenly the story broke that this doctor in Flagstaff, Arizona at NAU was the person working on this for the FBI in his lab. Within hours, we had news trucks from agencies around the world parked outside the building with reporters all trying to get in to talk to Paul. So we had all kinds of security immediately put in. We had police on guard duty 24/7. So many people were trying to get in to interview him. And we had FBI agents in lab coats wearing guns all over the laboratory saying, what can we do? We got to do this. We got to do this. What do we do? What do we do? And at that time, it got so crazy. Paul did not have someone to deal with all of the computer issues and all of the security that was all based on computer security models. And he finally came to me and said, I need to hire you. I need somebody to deal with all this stuff because we have no idea what we're getting into. We are a biosafety level 3 lab that deals with tier 1 select agents. Select agents are pathogens determined by the US government to be especially deadly to people, animals, and crops grown in America. We were actually the lab. We received blood and sputum and samples from the first victim in Florida of the Amerithrax letters. And before he died, which was 2 weeks after we received the sample, we had already distinguished it as Ames strain. So we knew it came from a laboratory and it wasn't a naturally occurring strain of anthrax. There was really very little genetics research on single cells and pathogens. This came from a mixture of different anthrax strains because when we were able to grow it on a plate, there was several odd-looking colonies of anthrax, not just one distinct formation of colonies. We were finally able to isolate the Ames strain that was in this mixture, and it turns out it matched a laboratory strain from USAMRIID, which is the United States Army Infectious Disease Research Center, and that was from a vial under the ownership of Dr. Bruce Ivins, who actually had worked on anthrax vaccine. In order to make it more difficult for people to figure out where this came from, he mixed it together with the wild strains, basically grew up colonies, killed them, let them sporulate, and then released the spores in these envelopes all over the East Coast.